Hello, good afternoon. Thank you very much to the organizers for the kind invitation to share our work in this very exciting meeting, in particular to Dr. Alejandra Medina, who invited us. Today I'm going to share with you some data we have collected in the laboratory looking at chromatin spatial organization during erythroid differentiation. We are located at the Institute of Cellular Physiology, UNAM, in Mexico City. So as you might be well aware, the chromatin compaction state is a very important regulatory layer for all DNA metabolism, including transcription, replication, recombination even, so that the accessibility state of a given sequence of DNA has the ability to modulate these processes. In particular, different compaction levels of chromatin have different nuclear dispositions within the cell, and normally heterochromatin or very compact chromatin, it's located towards the nuclear periphery, whereas euchromatin or more accessible chromatin it's located in the nuclear interior. Heterochromatin formation and maintenance is very important to sustain cellular homeostasis, so that heterochromatin regulates key events like for instance gene expression, as is a very repressive compartment of gene expression. It also it's also involved in silencing transposable and repetitive elements, providing those genomic instability and regulating it. And more recently, it has been well described that heterochromatin has an important role in organizing chromatin and providing it stability, so that when heterochromatin maintenance fail, nuclear architecture is disrupted and gene expression is altered. So during differentiation and maturation, there is a considerable gain in heterochromatin from the pluripotent cell to a precursor cell to a terminally differentiated cell. There is a considerable gain in heterochromatin. And we know quite a bit about how genome is organized in higher resolutions in pluripotent stem cells and early differentiated cells. However, far less is known on how the genome is organized in a terminally differentiated cell that contains a lot of heterochromatin. So when we talk about the different scales of genome organization, what we mean, and that of course you might be well aware as well, is that the genome is not randomly organized inside the nuclei, but chromosomes occupy territories, and in the subchromosomic scale, chromatin is organized in compartments, which corresponds mainly to euchromatic and heterochromatic regions of the genome that are binding each other and not are binding, euchromatin is binding euchromatin, heterochromatin, and the two of them are not communicating a lot. And then at the megabase scale, we can see that the chromosomes are organized in topologically associating domains, or TADs, which contain regulatory elements within them that are communicating through loops. And this provides, these topologically associating domains provide a spatial isolation for these regulatory elements, so that there is no incorrect crosstalk between regulatory elements in the genome. So as I mentioned before, there is not a lot of information on how all these different scales of organization look in a terminally differentiated cell that has a lot of heterochromatin and less gene expression. So we set out to investigate this using a very extreme cell in which this happens, which is the mature erythrocyte. So erythropoiesis is a process by which the erythroblast nuclei starts to condense, the heterochromatin starts to increase, and the whole genome and nucleus is compacted, and ultimately in mammals the cell nuclei is expelled from the cell, so that the mature erythrocyte is effectively a bag of hemoglobin that circulates throughout the bloodstream. However, this is not the case in the rest of the vertebrates, including birds, which is our model system, so that the process of differentiation occurs more or less the same way as in mammals, but the mature erythrocytes retain their nuclei. So this provided us with a unique landscape to look into chromatin 3D organization in this very heterochromatic compacted cell nuclei. So to do this, we studied the erythroblast stage of erythroid differentiation, and we looked at embryonic and adult red blood cells, two different states of mature erythrocytes. So we did this looking at chromatin accessibility, transcription, and 3D chromatin organization. And this is the outstanding PhD work of Andrés Penagos and also Sherlyn Claudio in the laboratory. And it's now a preprint at your disposition in bioRxiv. So first of all, we decided to look at chromatin accessibility through ATAC-C. So ATAC-C, you might also know this technique. So it measures chromatin accessibility as ATAC-C does, but in this case, the adapters that are loaded on the TN5 enzyme have a fluorophore, so that when the enzyme fragments the genome, introduces the adapter, and one can image where the euchromatic regions of the genome are located in the nuclei. So we did this in our different cells of study, and here you can just see a general picture of how it looks. So this is TAPI, this is the ATAC-C signal, and this is a merge, and we built this reconstruction, these images, by taking the signal that it's higher above the mean in both channels, so that we can study where the more accessible and the more inaccessible regions of the genome are located. So first of all, we saw, as we expected, that the general accessibility of the genome is decreasing in the red blood cells from the erythroblast to the red blood cells. We always use a fibroblast as a non-erythroid cell control. And we can see this very clearly by ATAC-C mean intensity, the percentage of the nucleus that is enriched with the ATAC-C signal, and the ratio between the ATAC-C and the DAPI. So indeed, accessibility is decreasing in the mature erythroblast compared to the in the mature red blood cells compared to the erythroblast. When we looked more closely at where the highest, the more accessible regions of the chromatin are located in these different cells, we could notice that in the adult red blood cell, the euchromatin is going towards the nuclear periphery, which is a particular distribution and a very unique one. And as I told you before, normally the heterochromatin is the one facing the nuclear periphery. So we wanted to measure this very systematically. So we calculated a correlation score of the signal, and we could see that indeed the red blood cells, the adult red blood cells, have a negative correlation to the distance from the nuclear rim, indicating that the majority of the signal is located toward the periphery, as you can see here. We confirm this by quantifying the signal along the diameter of the nuclei. And again, you can see here in blue how the signal is higher at the periphery in adult red blood cells, and then it decreases, which is the contrary of what can be observed for the rest of the cell lines, confirming that indeed the euchromatin in adult red blood cells is localizing towards the nuclear exterior. So we wonder if this phenomenon is due to changes in lamin contents in the cell, and we could see that measuring lamin B1 and AC, that indeed lamin AC is lost in the adult red blood cells, and this is not the case for lamin B1, so that the loss of a lamin AC could be the reason behind the detachment of heterochromatin to the nuclear periphery. So next, we decided to have a look at transcription in these cells. And for that purpose, we did a total RNA-seq analysis in fibroblast, erythroblast, and both embryonic and adult red blood cells. And first of all, I want to show you here, when looking at the RNA in these cells, we could see that indeed the ribosomes were almost undetectable already in the adult red blood cells, but we can still see an accumulation of a small RNA molecules. So we were very intrigued on which are these, and wanted to have a closer inspection of these molecules. So we saw that there are thousands of genes still expressed in the red blood cells. And here I'm just showing you an example of some of the genes that are still expressing, like the globin genes here very robustly in both embryonic and adult red blood cells. So when we analyzed all of these different transcripts, what we could see is, well, first of all, there is a still active transcription in the red blood cells, as you can see here, by uridine incorporation, because we weren't sure if this was leftover RNA or if indeed there was still active transcription. And you can see here that there is still active transcription in these cells. And when looking at the distribution of the different transcripts that are present, we could very interestingly see that in the adult red blood cells, there is a redistribution of the transcripts that are present, so that there is an increase in miscellaneous RNAs and snow RNAs, which is quite different from the rest of the cell types. And now, come back to this a bit later when describing which type of RNAs we found in these categories. So we next look also at accessibility, but now at higher resolution through ATAC-seq. And we found that there is still accessibility, as we could see from the ATAC-seq, and there are quite a lot of peaks present still in the red blood cells, both embryonic and adult red blood cells. And when looking closer in the genome to where these peaks are located, we could see that in quite a bit of instances, we saw accessibility located at gene promoters, which are of genes that are not being transcribed, as you can see here and here, for the embryonic and the adult red blood cell. So when we quantified this genome-wide, we could see that in general, the accessibility peaks in the red blood cells fall into promoters of genes that are not being transcribed, which is completely different of what we can see for the fibroblast and the erythroblast, in which the majority of the peaks are there in genes and promoters that are active. And we confirm this here by plotting the actual signal of both the accessibility and the RNA. And you can very clearly see how here the accessibility is maintained, but the transcription is decreased very robustly in contrast to the fibroblast and the erythroblast. So having seen this, we wonder if the gene promoters in the red blood cells are occupied by the RNA polymerase. And what we could see, first of all, is that when looking at the RNA pool 2 phosphorylated in serine 5, we could see that there is still quite a lot of signal in both the embryonic and the adult red blood cells. And very interestingly, in the adult red blood cells, the disposition of the post-polymerase corresponds to the signal of the ATAC-C, suggesting that those peaks are indeed occupied by the post-RNA2 polymerase. Moreover, when looking at these miscellaneous RNAs from which I talked about, you could see that C7sk, which is a very important transcript involved in the posing of the polymerase, is very prominently expressed in the red blood cells, and it's the large majority of these miscellaneous RNAs. And also we could detect the presence, for instance, of exim, which is a protein also involved in polymerase II posing. We then went to confirm if some of these peaks that we could see at promoters of the different genes are indeed occupied by pol 2 serine 5, as suggested by this immunostaining. And as you can see here, for instance, for these 3 genes which are basically have an accessibility peak at their promoter, but are not being transcribed in red blood cells, you can see how indeed the signal in the red blood cells is pretty abundant, so that indeed this promoter seems occupied by RNA pol 2, and this is just a negative control. So finally, we decided to have a look at the 3D chromatin organization at high resolution through Hi-C, and I won't go through this protocol, as you might be well aware of how it works, but effectively one ends up with all the chromatin contacts of the, that different genomic regions are undertaking in a given moment in the cell, and with this we can build 3D contact maps of all the genome. So by doing this in our different cells, straight away we could see a clear difference between the blast and the red blood cells, in which we could see this very strong signal at the compartment level in the Hi-C matrices. So when making the differential, taking the 2 signals and making a differential matrix of these 2, we could very clearly see that indeed in the red blood cells, the long-range contacts are far more abundant than in the blast, and in conversely in the blast, the short-range contacts here near to the diagonal are very increased, and the difference between the embryonic and the adult red blood cells is not very big. They are pretty similar at this scale. So we went to look at this a bit more systematically to see if indeed this hyper-compartmentalization that we could see was there genome-wide. And in this is something we observed. And interestingly so, what we could see is that there is an invasion of the B compartment on the A compartment in the erythroblast and in the red blood cells and in the embryonic and the adult ones, so that heterochromatin is pretty much invading euchromatin in these cells, as you can see here by the increase in the yellow signal in these tracks. When quantifying this genome-wide, we could indeed see that the red blood cells have more B compartment, which corresponds to heterochromatin. When looking a bit closer into these high symmetrices, we could see also that apart from this hypercompartmentalized pattern, the presence of topological associating domains was less evident in the red blood cells compared to the fibroblasts. So we again went to quantify this in a systematic manner using a metric to call topological associating domains in these genomes. And we could see that, first of all, there is a massive loss of domains in the embryonic and the adult red blood cells compared to the erythroblast. As you can see here in these images, the loss of these of all these structures. However, we could still see small domains, which we named mini domains, in these erythroid genomes. And when quantified this in a genome-wide manner, we could see that indeed the number of domains is pretty much is decreasing robustly in the red blood cells, and both by counting them and by looking at the genome coverage of these structures. And we could confirm that the domains, the tags that are left, are smaller, significantly smaller in size in the red blood cells compared to the erythroblasts. So we wonder, okay, so how these mini domains relate to the accessibility and the RNA-seq signal that we have measured before. So we integrated these different analysis, these different data sets, and very interestingly, we could see that the majority of the mini domains in the red blood cells are organized around post-gene promoters that don't have transcription. So we classify the domains in these 4 groups, having a post mini domain with accessibility and no transcription, then a normal open and transcribed TSS associated to a gene that is transcribing, then a class 3 in which you have a combination of both a scenario, and a class 4 in which the TSS is completely closed and there is no transcription. And as you can see here, in the red blood cells, as I mentioned, the majority of the mini structures correspond to class 1 and class 3 domains, so that mostly these domains are organized around post promoters in this genome, which is very different from what we can see in an erythroblast, in which the more common domains are structured around active genes. So when looking a bit more in detail, which type of genes are present these post-state structured in mini domains, very interestingly, we could see that most of these genes fall into RNA metabolism, transport, processing, etc. So that we think that this genomic structure is providing a post-structural state, transcriptional structural state of this genome, so that upon certain stimuli, they might be able to mount a significant transcriptional response and start transcribing. So just to finish, I've shown you that from the erythroblast to the embryonic and the adult red blood cells, there is quite a decrease in transcription, but certain genes are still being transcribed, and that chromatin accessibility is also decreased, but there are still regions that are accessible and important in these genomes, and that at the level of chromatin domains, there is a loss of these structures. However, small mini domains are organized around post gene promoters. Also, we could see this hypercompartmentalization of these genomes due to the invasion of the heterochromatin to the euchromatin. And finally, we could see that these accessible regions are now located towards the nuclear exterior, present an inverted chromatin architecture, so that these mini domains, these post promoters, are presumably located towards the nuclear exterior, and this is also due to the loss of lamin AC in these cells. So with that, I want to stop here, and I want to thank Andres and Charlene, which are the key students working in this project. Also, our Stevenson has participated quite a bit, and I also want to thank all the budget agencies, PAPIT and CONACYT. And I also need to thank, and sorry, it's not here in this slide, but Dr. Cesar Pot, which has been of great bioinformatic help in this project, and Dr. Xin-Hui Chen, who donated the ATAC, the TN5 enzyme linked to afluorophore, so that we could perform the ATAC-C experiments. And with that, I thank you all, and I'm happy to take questions.